This presentation will cover some of the strategies for automated brain morphometry analyses. As described in previous presentations, some of the common steps that are included with morphometric analyses as well as other types of neuroimaging research include skull removal, bias field correction, image registration, and segmentation. Another critical step, obviously, is the statistical analysis, which is dependent on the type of question that we're asking. A number of tools have been developed and are available for researchers to analyze the brain imaging data, including for morphometry, and they can be found through NITRIC, which is a web-based resource for neuroimaging tools and sponsored by the NIH. FSL offers a wide variety of different tools for structural image analyses. These include FAST for image segmentation, FIRST, which does subcortical structural segmentation. We'll also talk a little bit about the FSL voxel-based morphometry analysis, but then there are other tools as well. Here are some pictures illustrating some of the functionality of the FSL FIRST, shown on the right, which provides probabilistic templates for segmenting subcortical structures deep within the brain, and it works fairly well. In addition, they have a tool called FSL NAT that includes FSL FIRST, but also includes such functions, including brain extraction, segmentation of gray matter, white matter, and CSF, and hemispheric labeling. Voxel-based morphometry fits within the broad category of computational neuroanatomy, where we take a set of images across individuals, and typically it starts out with spatial normalization, and then with that we can either process the normalized images through a pipeline called voxel-based morphometry or examine the deformation fields, and those techniques are typically called either tensor-based or deformation-based morphometry. Probably the most common technique, however, for analyzing morphometric data with MRI is called voxel-based morphometry, or VBM. And FSL has a tool for automating much of this processing. So here's an illustration showing the comparison of two groups of images on the top and bottom rows. So the original images are first skull stripped, then segmented, and then co-registered into a template space. The gray matter segmented images are then smoothed, and then statistical testing is performed at each voxel location. So for example, if we wanted to compare the top group versus the bottom group, the highlighted regions on the right show where there are significant group differences. And this is done at a voxel by voxel basis. And the main steps associated with voxel-based morphometry are to first segment the images, then spatially normalize these segmented images to some sort of standardized brain template. We then smooth the normalized data to generate a map of anatomic density, and then do statistical analyses at the voxel level. And so some of these steps are more directly outlined here. So here we're showing the original image on the left after skull stripping in the middle using brain extraction, and then the image on the right after fast segmentation generating a map of the gray matter density. The gray matter density images are then warped to a standard template um, using, in FSL, they use FNERT, and this is what the population averaged template looks like. If you're comparing two populations, they recommend that you have the same number of participants in each from each sample to create the template so that you don't bias it towards one population or the other. You then take your individual gray matter density images from each individual and warp that to the template. And the one process that needs to happen as a function of this warping is that you need to adjust for the expansion or compression of the deformation. So for example, if you had a really narrow region of gray matter that you are expanding in the normalized space, you need to adjust that gray matter density for that degree of warping. And just a brief review, so if we have an area of either protrusion or indentation in our brain, that's going to, when we warp it to our template, we're going to have a deformation field. And so we need to correct for that deformation in our gray matter density maps. And we do this using what's called Jacobian modulation. So if the image on the left, if we warp that to the image on the right, the area around the eyeball on the right is getting compressed down. And so that means that we need to scale the intensity of that eyeball signal according to that deformation. And that's done through the Jacobian. And the Jacobian is essentially describes the transformation or expansion and compression in 3 dimensions as a function of the overall transformation. And it's described by this 3x3 matrix. What we are for deformation-based morphometry, what we are, or for correcting this, what we care about is the Jacobian determinant, often just called the Jacobian for scaling. So using the Jacobian map, we can then use that to scale or correct the warped gray matter density image. So if we have an area where the gray matter is contracting, the gray matter density will increase. However, if we have regions where the gray matter is expanding, the density will decrease. The next step, after all of our gray matter density images are warped to the common space, is to smooth with a Gaussian filter. So this essentially blurs our images. And typically we use on the order of about 5 to 10 millimeters. The purpose of this is to first remove noise, but then also, since brains are not identical, we want to minimize any kind of spatial misregistration. And so the blurring helps to accommodate that. And then once we've have all of our segmented, warped, and smoothed images, we take and do our statistical testing at the voxel level. And we can do a variety of different kinds of analyses, and we'll go over some of those later in the semester. But for example, we could compare groups or try and model the density versus a continuous variable. So for example, how does gray matter density change as a function of age or IQ or as a function of disease severity? Some of the advantages of voxel-based morphometry are that we are analyzing all of the regions at once. It does not require an a priori definition of specific regions of interest. And we can also detect differences within small subregions of a region of interest. So for example, rather than testing the entire hippocampus, we might find differences that are constrained to the tail of the hippocampus and not the head. It is a controversial approach, however, and that's partly because the segmentation is and blurring may make it difficult to disambiguate the more complex features of the cortical gray matter. So whether a region of gray matter is associated with folding or gyrification, it's not able to disambiguate all those kinds of patterns. The two main software packages available for doing these types of analyses are FSL, which has the FSL VBM, and SPM, which is based in MATLAB, and was the first tool that became available for doing voxel-based morphometry. Tensor-based morphometry is based upon analyzing the actual deformation fields from the spatial normalization. And you can either use the Jacobian of the deformation fields, or if you want to try and analyze the full 3D deformation, you can use a multivariate analysis of the actual deformations. That's very rare, although the Jacobian analyses are growing in terms of utility. Another way of analyzing morphometric properties of the brain is using a tool called FreeSurfer. And essentially this has some commonality to the techniques that we've described previously. It takes our T1 weighted images, does skull stripping, then some initial segmentation of gray matter and white matter, some volumetric labeling of deep gray matter structures, intensity normalization, and then it reconstructs the gray matter surface on top of the white matter. You can then take this gray matter surface, register that to a surface-based atlas, and then do testing on the cerebral cortex. And this is called surface-based morphometry. And we can look at things like look at measurements within either at the surface vertex base or within specific gyral patterns. And FreeSurfer is one package that does this. SPM has a tool now called CAT12, and ANTS also has a tool for estimating cortical thickness. In addition, you can try and measure properties of cortical surface area and local gyrification. Other types of analyses that related to morphometry are to try and characterize the actual shape of either the brain or substructures. And so here's an example of a study that is analyzing shape differences in deep brain structures, namely the caudate, putamen, and thalamus. And they're using what's called spherical harmonics to try and model the surface of these structures and compare the differences in the actual shape of those structures. Some of the predominant challenges associated with morphometry studies include poor image contrast. So for example, in infants, the contrast between gray matter and white matter is quite poor. And that's because the brain is not fully myelinated yet. And myelin is one of the primary mechanisms for contrast in structural images. Also, if the imaging parameters used for a study are suboptimal, you may not get very strong contrast between gray matter and white matter, which will limit the ability to segment imaging structures. Image motion is a significant challenge, particularly in young or intellectually impaired populations, as well as the aging. And in imaging research, we can't use sedation. So cases where the subject moves, we will get a lot of blurring and ghosting in the images. And so those images are not really usable for quantitative studies. And then finally, studies that look at pathology, or if you have cases with unintended pathology, that will mess with the overall ability to do accurate segmentation and spatial normalization. So in summary, morphometry is a class of techniques that we can use to estimate and compare the size and shape of the brain and substructures. And the key steps associated with these sorts of measurements are spatial normalization, image segmentation, and voxel-based morphometry, which allows us to do a local comparison of anatomical size and shape. Some of the other considerations are analyzing images with multiple contrasts at the same time, how to analyze lesions, both the impact in terms of directly assessing lesions, but also assessing the effect of lesions on other structures. Cases where we have poor or variable contrast or just generally poor image quality. And really, the field of machine learning and deep learning is really driving a revolution in terms of providing newer and more robust techniques for doing a lot of these sorts of processes. And so I anticipate in the upcoming years that a lot of these strategies for analyzing structural morphometry will be significantly changed. Thank you.